Okay, good afternoon, everybody. Thank you all for joining us this afternoon. So on behalf of the Biochemical Society and Portland Press, I'd like to welcome you all to Biochemistry in Focus webinar series. Today, we're going to hear from Dame Professor Janet Thornton, who is a senior scientist at EBI. But before I introduce Janet, I will tell you a little bit about the webinar series. So the webinar series has been running through the pandemic, I suppose, maybe just a little bit before. And it's a series of free-to-air webinars on topics of interest across the breadth of molecular biosciences. And so it's open to everybody, and we're welcoming suggestions for talks in the future. But today, it's really great pleasure for me to introduce Janet Thornton to everybody, though she probably doesn't need a huge amount of introduction, because Janet is one of the foremost molecular modelers and computational biologists, and it's really for that role that we're going to listen to her today. She was awarded the 2021 Biochemical Society Award for specific work in the area of computational and molecular biosciences. So Janet is currently a senior scientist at the EBI, at the Sanger Institute and the Genome Campus in Cambridge. She was the director of the institute until I think about 6 years ago, but she continues to work very prolifically in the area of molecular modeling and enzymology. Amongst her many attributes and strengths, Janet has published one of the most highly cited papers. You'll probably know about ProCheck, which I think has been cited over 24,000 times, I think. And she, not only with that paper, but with a series of other papers in related areas, she has become one of the foremost computational bioscientists over the years. So with that, I'd like to introduce Janet to you this afternoon. She's going to speak for 40 minutes, so she tells me, and I'm going to butt in at 35 if she looks like she's going to go over. So it's a really great pleasure to introduce you, Janet, and congratulations on being recipient of this award. Thank you.
Okay, can you see my screen, Colin?
I can indeed, yes, thank you.
Brilliant, thank you very much. So first I should say good afternoon to everyone. It's a pleasure to be here. Secondly, I'd like to thank Colin for his kind introduction. And thirdly, I'd like to say how much pleasure it gives me to be the recipient of this Biochemical Society Award. The BioChemSoc has been very important, I think, in bringing together biochemists around the UK. It always has an exciting programme of lectures and conferences, and it's really a pleasure for me to be nominated and given this award. So today I'm going to tell you about computational enzymology, the structure, function, and evolution of enzymes. And of course, I'm sure you all know a lot about enzymes, but just to give you a bit of a historical perspective, of course, they've been used for enzymes from yeast and fungi in food productions since 7000 BC. They're used for making cheese, for making bread, and for making alcohol. You can see here in Japan making sake with enzymes. But the actual molecules were discovered in about the 1800s, and catalysis was first given a name in 1835. The concept of an enzyme, an individual molecule that performs these chemical reactions, and the fact that it was cell-free fermentation was discovered later that century. And during the 1900s, many different aspects of enzymes were characterized. Perhaps most importantly, the metabolic maps, pathways that you again will be familiar with were identified. The idea of curly arrow diagrams were invented by biochemists to explain the mechanisms of enzymes. And of course, during this time, structures were determined and sequences for the first time. And now, in the 2000s, we really do have enzymes galore. Most genomes have between 25 and 50% of their proteins being enzymes. We have millions of sequences, thousands of structures, and we know that these structures perform many different reactions, of which you can see one here. Quantum chemistry methods are used to analyze enzyme mechanisms, and indeed recently a few successful enzymes have been designed. But despite all of this knowledge and work, there's still many outstanding questions. For example, how do enzymes really work? How does this green chemistry allow these reactions to happen? How does enzyme function evolve? Can we predict transformations and mechanisms? And can we predict specificity and function from a sequence and structure? And I'll come back to some of these issues. And of course, these are really good academic, challenging questions. But actually, there are many applications for which solving these questions would be important. And of course, enzymes are very important in biotechnology, in medicine, and in agriculture. They're important for drug design. They're important increasingly for designing new enzymes. They're important when we try and design new pathways. And of course, the green chemistry that you can see here has many different powerful applications. And wouldn't it be great if we could really find some plastic processing enzymes that would help to remove some of the waste that has been generated? And of course, they are relevant to medicine. If we look at drug targets, in fact, about 54% of all drug targets are enzymes, of which 75% are human enzymes. And indeed, if we look at novel drug targets that were identified in 2020, then we find that 5 out of 13 of these are indeed enzymes. So today I'll talk about really 3 aspects. First, I'll tell you a little bit about the database that we've created to capture information about enzyme active sites and their mechanisms. Secondly, I'll talk about the evolution of enzyme functions in the study of enzyme families. And lastly, I'll give you 3 very short descriptions of some ongoing work in my lab to do with enzymes. And of course, in the lab, our goal is to capture, to analyze, to validate, and ultimately predict enzyme mechanisms, and to discover how enzyme function in a family changes during evolution. Before I start, I'll just tell you about EC numbers. These are enzyme permission numbers. They're quite beloved of chemists and computational biologists. They have been, it's a 4-digit hierarchical classification of enzyme reactions. So if we look at this reaction at the bottom, it involves the small molecule that is transformed by the enzyme. And this classification of enzymes depends only on this reaction. It doesn't depend on the protein at all. It is purely a description of the reaction performed by an enzyme. There are 4 numbers. The first one is the class, and then subsequent numbers get, if you like, less and less important from the chemical point of view. So you can see any enzyme, in this case, this enzyme is described as an EC 3.1.4.11. And the main classes are shown here. I should say that I've told you all about the data that is available. There are several databases, public databases, which are really useful and store a lot of the information about enzymes. However, back in the early 90s, 1990, we decided that we would start to look at enzymes in terms of their mechanisms and the proteins that perform this catalysis. Now, at that stage, we had the PDB, the Protein Data Bank, that had the coordinates, but there was really no information in the PDB about which were where the catalytic site in an enzyme was, or which were the residues that were important. So we decided to gather this data, and in fact, every summer we have had 3 or 4 students come to go through the literature and extract this information from the literature. And this just shows the information that we store about enzymes. We store the reaction and the EC number and the name. We store information about the proteins that perform this reaction. So we have a protein with a defined sequence and structure. We also have information about the cofactors, whether it's an organic or a metal ion, and the catalytic residues that are responsible for defining the active site and for performing the catalysis. But as well as that, we have also captured the mechanism, how these famous curly arrow diagrams, how an individual enzyme actually performs this catalysis. So this is the basis of trying to understand how this green chemistry works. And this information we now have, I think I showed in the previous slide, we have actually now we have a few more than that, almost 1,000 enzymes in the MCSA, and we have mechanisms for about 700 of those. So let me now move on to using that data. We wanted to look at the evolution of enzyme function, because it's very clear that an enzyme family during evolution in different organisms and different environments will evolve to have members of this family which perform different enzyme functions, perhaps 3 functions in this case. And I have an example here looking at one particular superfamily that I'll call PIPDE. This is a very large superfamily, many sequences, many species. We have at least 26 in our genomes, and also we have information about their structures for 39 of the, 39 exam structures are in the PDB, and this enzyme superfamily also has different functions. So it's not just a phosphatidyl inositol phosphodiesterase, it does other things as well. And in fact, there are 18 different functions, or there were when we did this analysis, which is now some time ago. And you can see this is the reaction, the 3,1,4,11 reaction for one member of the PIPDE superfamily. And you can see the substrate involves a ring structure, phosphate, and you can see the green here, and the enzyme cleaves this bond here. And this enzyme actually needs magnesium as a cofactor to work effectively. Now, we can take all the sequences for this superfamily and generate an evolutionary sequence tree. And in this tree, what I've shown is the EC numbers that capture the function of these enzymes for all the members that belong to this sequence family. And you can see those EC numbers change. Most often they change in the last digit of the EC code, 43, 44, 46, 4, 46, 14. But sometimes they also change in the class. They become a lyase instead of an esterase. And so we decided to look at this family, and you can see that there are structures for 3 members of this family that are shown with the pink and the blue. And those members, they all look very similar. They clearly are homologs, but their names, when you look at them here, you can see we've got sphingomyelin phosphodiesterase, or we call that SPD, the phosphoinositide phospholipase, PLC, and the I can never pronounce these, phosphatidyl inositol diacylglycerol lyase, PDL. So we have these three different structures that are similar, but certainly not identical. And when we look at the reactions that they catalyse, I've drawn them here, the SPD, PLC, and PDL. And on the left-hand side, you can see the substrates. And the first thing you notice, this is the bond here with the arrow that's cleaved. So that part of the molecule of the substrates is very similar. But actually, this part of the substrate that you can see for the sphingomyelin is a very long extended chain, whereas these two actually look very similar, but they both have the ring structure. You'll also see that this enzyme, the SPD, uses magnesium, the PLC uses calcium, and the lyase, the PDL, doesn't have any metal cofactor. And so, first of all, we looked at this change of substrate. This is SPD here, where we've got this substrate being transformed and this bond here cleaved. And you can see this is the active site of the SPD, and it has a magnesium right at the center. If we compare it with the phosphoinositol, the PLC, phospholipase, we can see these are the two. First of all, the substrates are very different. There's a change in substrate. They both have the metal ion here. And when we look at their binding sites, their catalytic sites, I should say, not the binding sites, we can see that they really superpose very well when we're comparing the two. And the magnesium and the calcium lie in almost the same place. They overlap completely. But when we then look, so in this case, we have a change of substrate because the shape of the binding pockets that you can see here are very different. You can see one is very open for the PLC. For the sphingomyelin, it's a much deeper tunnel, and that long side chain protrudes sort of out of the enzyme. So we have what has happened is that the enzyme has evolved a different shape binding site, and those bind very different ligands. But now we look at the comparison with the PLC with the the PDL. Okay, so here we have the PLC that we've already looked at, and we can see this binds the calcium. You can see it there. When we look at the PDL, there's no metal binding site in this enzyme. We can compare them, and some of the catalytic residues superpose quite well, but we have a very different residue here. And in fact, this is an arginine, and that arginine protrudes into the binding site where the calcium or the magnesium would normally bind. And you can see this is not possible, and therefore for the PDL enzyme, there is no binding of that cofactor. And when we look at the binding site, the sequences of these, and you can see them here, at this point, we have got a change in the amino acid sequence from an aspartic acid to an arginine, and that arginine, as we saw, precludes the binding of the calcium, and that means that one part of the reaction cannot occur, and so this becomes the lyase instead of the esterase. So this is evolution. The change from asp to arg leads to a loss of that metal binding. So we decided we would look at these sort of changes, and we did a lot of computational things to analyze many different sequences. And for each family, we generated what we call an EC exchange matrix. So in most cases here from this tree, you can see that the EC3, which is the hydrolases, become other EC3s, but with different substrates. Whereas when it changes to a lyase, then we get a change from EC3 to an EC4 or 4 to 4. So we can plot this EC exchange matrix for each of the families that we look at. And we've generated then a combined EC exchange matrix. Oh, hang on. Let me look at. So here we count of the number of changes in function that we observe. We count how many of them are between the major classes. And what you can see is that most exchanges occur within a class. So hydrolases become other hydrolases just with a different substrate. Or transferases become a different sort of transferase with a different, perhaps a different atom or part of a molecule transferred. And so from all of this analysis, and we analysed 276 families, we can see that in evolution, 89% of the changes are changes in substrates. That's the most common, and it's kind of the easiest thing to do. If we look at changes of reaction chemistry, they do occur. And in fact, all sorts of changes are observed. None of these off-diagonal elements are zero. So they all occur, but they occur much more rarely. So during enzyme evolution, changes to substrates occur much more frequently than changes to chemistry. And so we can plot, and this is work from Sergio Martinez, who was a PhD student in the lab. He looked at isomerases in great detail and found examples in the database where isomerases have become liases, oxidoreductases, transferases, hydrolases. And the case that we saw, liases, we saw an exchange from, it was from oxidoreductases. No, it was from liases, hydrolases to liase before. And we can plot these for all the enzymes that we know. And I really love this plot because to me it illustrates the power of evolution to create enzymes with new functions. It's fantastic. An enzyme can evolve to do something not totally different, but definitely different. And in fact, most of the enzymes we see today are the result of these variations within families, rather than the invention of completely new enzymes. And I should mention, just in passing, that this is the kinases, which is a huge family, and that sometimes enzymes evolve not to produce different functions, but to be non-catalytic. And in the kinases, many of the kinases, all the blue ones shown on this evolutionary tree, are non-catalytic. So the evolution to losing activity and doing other things is also really important. So that concludes what I'll say about evolution, except that everything that we look at, of course, everything in biology only makes sense through the lens of evolution. And I'll just describe three short aspects of work in my lab. The first is by a PhD student, Yannis Riziotis, and he's interested in the flexibility involved in enzyme catalysis. And so as a starting point, we asked, are enzyme active sites and catalytic residues flexible? And the idea was to use enzymes with multiple structures in the PDB to investigate their flexibility. Oops, sorry. And what he has shown, and what is known, but it's good to have it all collected together. When you look at overall sequence similarity, we learned long ago from Chothier and Less that as the sequence similarity of two proteins decreases, then their overall C alpha root mean square deviation between the structures increases. So here you can see two superposed structures. There is broadly, as the sequence gets more different, the structure gets more different. And this is what you see for the overall fold of the protein. However, If you just focus on these active site structures and do the same sort of analysis, but focusing on the C-alphas of the catalytic residues, what you see is that now, although the sequence similarity can be very, very low, in general, the structural similarity is very well conserved. And these active sites really are a little bit like rocks in the center of the enzyme. They really don't change during evolution. Their structure, in general, not always, but in general, their structures are conserved. And so we've been looking, and he's now completed an analysis. The paper actually is in bioRxiv of 307 different families. And we've looked at how these families change their structures, what sort of flexibility you get in these active sites. And this is just a summary of the results that he finds. We find some sites, in fact, the majority of these 4 classes, are inherently pretty rigid. Those catalytic residues don't move, they retain the same structure and conformation. There are others where the active site is much more flexible, and all residues in the active site really are quite flexible and adopt different structures. There are other enzymes, and that again, a large chunk of them are in this sort of state. Other enzymes have 2 states. They're open when there's no ligand bound, and then they close in on the ligand. So there are just 2 states, in which case you end up with a root mean square deviation plot that looks something like this. And then there are others again that seem to vary all over the place, and they're extensively varied. So Yanis is carrying on with this, and we're trying to understand more about the evolution and how flexibility is important for that evolution. Another project, which is a project of a very talented postdoc, Antonio Ribeiro, is to try to predict enzyme mechanisms using an informatics approach. So we know that quantum chemistry is needed really to predict mechanisms, but we'd like to use the information that we've got to suggest possible mechanisms. So we know that if we have a reaction like this one here, which is glutamate racemase, I think, then there are different steps involved in that reaction. First of all, the proton is stolen by the aspartic acid from the cysteine. The cysteine then attacks the glutamate, one of the proton on one side, and then that proton is transferred and a proton is gained from the other side, if you like, onto the glutamate in the middle. So this is the mechanism. The question is, could we create an algorithm that, given an active site and the reaction of an enzyme, to predict possible enzyme mechanisms based on the mechanistic information stored in MCSA. So could we take this active site information with knowledge of the reaction and predict what this mechanism might be? Based on a knowledge-based prediction, based on all the mechanistic information that we've got stored in the MCSA. So this project is now 2 years old. Antonio started 2 years ago to try and do this. It's been an interesting journey. It's not straightforward, but actually he's now got to this position where he can predict a whole set of configurations of the enzyme. So he's parsed the data stored in the MCSA and extracted rules that govern the enzyme chemistry. Then he's created this algorithm, so this is now done, to explore possible paths. He's created a view that shows the predictions, and now we're in the process of validation. And I think within the next few weeks, he will complete this. It's not easy, we don't get it all right, but we've certainly made progress in this. So this is very exciting. And then the last aspect that I'll talk about is towards function prediction using AlphaFold models. So I hope you are familiar with the AlphaFold prediction method. This came out the first results from this were published in December 2020. The paper was published during summer in 2021. And basically, this algorithm was shown to be extremely good at predicting the structure of proteins, the coordinates of the protein from a protein sequence. It does it better than any other algorithm in two ways. It's very good at predicting accurate structures based on knowledge in the PDB for homologous proteins. So homology modeling, it's very good at. It also sometimes works for predicting de novo structures. And in the CASP, the critical assessment of structure prediction, this method made a huge leap. First of all, it predicted structures better, almost all the structures better than any other team. And secondly, the accuracy of the predictions was remarkable. So what happened as well, they published a paper on the human proteome, and they showed, just at a very high level, that although only about 38% of human proteins have structures in the PDB, for AlphaFold there are predictions for almost all the proteins. Although one should say that, in fact, they are often partial predictions, and the quality of the predictions for some of the residues is very low, the reliability, and those parts of the proteins should basically be ignored. Anyway, this approach was developed by DeepMind, which is a company based in, it's part of Google, based in London, and they really took the world by storm. This method to predict structure became Nature's Method of the Year in 2021. But furthermore, although they are a commercial company, they decided to make their predictions available for everybody. So they have collaborated with EMBL-EBI to create the AlphaFold database. It's open access, these models are available for everyone, and you can download them all via FTP. There have been 2 new releases since the beginning in July. One describing all the annotated proteins, Swiss prot proteins in UniProt, but also describing the proteomes for the structural proteomes of organisms involved in many neglected tropical diseases and those organisms responsible for antimicrobial resistance. And in fact, this database was launched in July and is very well used. Since it was launched, it's had over 368,000 users. And here you can see the countries that have been using it most. But now let's think about enzymes and the AlphaFold coverage of enzymes in human proteins. So we know in the PDB, we have about 11,000 non-enzymes and about 4,000 odd enzymes. So about a quarter of the structures for the human proteome in the PDB are enzymes. But if we look in the PDB for those sequences with no structures and no homology models, then we still find that there are 36 potential enzymes in the AlphaFold database. And so the question is, what do these enzymes do? And can we in any way find the function? And so we have been using these models to search for enzyme functions. And the way we've done it is to derive functional residue coordinate templates from the PDB. So here you can see the classic catalytic triad with the asp, his, and serine. And we can make, these are the catalytic residues. The MCSA, our database, has this information for almost 1,000 enzymes. So we can make templates for these different enzymes. And then we can scan each of the AlphaFold models to see if it provides a region that matches one of our enzyme templates, and we can highlight the best hits. And in fact, this approach was developed, well, a long time ago, more than 10 years ago, when during the era of structural genomics, when many structures were solved and we didn't know what they did. So we have a lot of catalytic residue templates. They're all basically 3 residues, so one catalytic site can generates more than one template. And here you just see a selection. So we use these templates then to search the AlphaFold model and ask, do we see this arrangement of residues, and will this tell us what these proteins do? So this shows one example where this was one example of a structure from AlphaFold. In fact, it's a human transmembrane protease serine 2. And what we found, of course, was that the catalytic template, the catalytic residues in this model overlap perfectly with our catalytic template. And so we are absolutely confident this is a serine protease. But we actually have all these templates. We also have templates for DNA binding, and we have templates for ligand binding. So we have lots of different templates to use. And when we scan about 300 of these protein AF models for which we have not previously had structures, what we find is that our templates, we find templates that hit for enzymes, for ligands, and for DNA. And we also have a slightly different method called reverse templates that has even more hits. So this is one way that we can use these new, more reliable models to try to annotate them with functions. And so just to conclude then, the future work that we still are involved in, we are obviously proceeding with our mechanism predictions and seeing how far we can get. I think it won't be all the way, but I think it will be helpful. We're analysing active site conformation, and in particular the flexibility and dynamics of that. And we're doing that by constructing a 3D library of these catalytic templates. Of course, I haven't spoken at all about enzyme repurposing and enzyme design. And that is really important, the subject of a whole nother talk. But really very interesting to use this knowledge that we have in the MCSA to try to design novel enzymes. And lastly, of course, we would like to understand in more detail how each individual family can evolve and develop new functions for enzymes. So I'd like to acknowledge the people who are currently in my group, the people shown here, and also many people who worked over many years with the MCSA, with the data. Gemma Holliday originally set up the mechanism or the mechanism data. Assad worked on developing tools to compare enzymes. Nick Furnham did the evolution work that I showed, and Sergio worked on the isomerases. And this is all a collaboration with Christina Rengo and her team at UCL. And our funders are mainly EMBL, but also the Wellcome Trust. So I hope I've excited you about enzymes and what they do and what we can understand about them. Because I really think that there's many things left still to understand, and that maybe some of you might work in a similar area going forward. Thank you.
Okay, thank you very much, Janet. That was a fantastic presentation. So I hope everybody can hear me. So really clear presentation. So we're going to open up the chat to questions. So if you have some questions for Janet, then please type them into the chat box and they will hopefully relay onto my laptop and I will be able to read them out. And while you're doing that, I'd just like to say a little bit more about the Biochemical Society Award. So as I mentioned at the start, Janet is the recipient for 2021 of the Biochemical Society Award. This is an award that the Society instigated in 2001, and it's given out every 2 years. And unlike many of the awards that the Biochemical Society gives out, this one is given out to scientists working in specific areas dependent on the views of the award committee and the trustees of the Biochemical Society. And in 2021, it was awarded, or it was nominations were sought from candidates who demonstrated outstanding contributions or applications of computational mathematical methods leading to advancing the understanding of molecular biosciences. And I think you will agree from Janet's presentation and from understanding something about her illustrious career that this is certainly an award that we almost could have written the citation for her before she was awarded it, but we didn't. I will tell you that we didn't do that. So anyway, it's a great pleasure to be able to award this to Janet. I would dearly like to have given it to her in person, in real life, but things being as they are, we obviously can't do that at the moment. The Society is moving back towards in-person meetings slowly as the pandemic hopefully wanes, and hopefully will continue to wane. Of course, we don't know, because we don't know the evolutionary path that the virus is planning for us, but there you go. So I would really like to have done this in person, but we're not going to be able to do it like this. So here is Janet's award, and let me say congratulations, and I would like to present this to you, Janet. This award will be winging its way to you in the post sometime very soon, so you'll be able to place it alongside all of your other awards, and I hope you'll think of it as being equally prestigious. So congratulations. So, you're very welcome. As I said, if people have questions, I think we have around about 10 minutes for questions, and I will try and field them. I'm working with 2 laptops and my eyesight isn't fantastic, so you maybe have to bear with me. I will look this way because that's where the questions are. So if I start, so the first question we have was from Brianda Das. And he said, why are housekeeping enzymes, why do they not change their characteristics so much?
Well, because they do the basic jobs, and nature is very good. If it's found a solution, it doesn't change. It just keeps, I mean, this is the amazing thing about protein structures. We know that during evolution, sequence changes all the time, but the structure and these catalytic sites in the core of the enzyme are totally conserved, because if they aren't for housekeeping enzymes, then you can't keep the house. The whole thing doesn't work. And so those residues are extremely well conserved. And although I talked about these changes in function, actually in the grand scheme of things, they are very rare. They don't occur, you know, most of the time. Thankfully, our enzymes are stable and they carry on doing the same job. And that job, as I say, is, you know, present in many, many, many species.
Okay, thank you very much. So the second question from Nicholas Hammer sort of almost reverses the situation. So could these prediction methods actually be used in reverse to help to design novel enzymes?
Well, I think that's at the back of my mind. Actually, I'm not a very good predictor, because I know how difficult it is to do these things. And so I always enjoy looking at how things work, what we know about them, and trying to evolve or make tools that will help us to annotate the enzymes. But actually, in my mind, it's like a car. if you understand how a car works, then when it goes wrong, you can correct it, or you can build a new car with something slightly different. But you need to understand the rules. It's no good just to sort of have one example. It's helpful to understand. And I think it's also helpful to understand how evolution works, because then you can take an old enzyme and design a new one that looks very like the old one, but does something slightly different. So it changes its substrate. That happens all the time in evolution. And it's something I'm sure we should get to the point of being able to predict. And I think it's an area, I didn't talk at all about the AI and the DeepMind methods, machine learning methods, but there's no doubt in my mind that these will become very powerful for designing new enzymes.
So the next question from Claire Coupland is, have you tested some of your approaches on internal membrane enzymes?
So where we have structures, we can look just as easily at membrane-bound proteins as not. But as you know, the number of membrane protein structures for enzymes is still quite small. So we don't have a lot of information. And very often, in looking at the flexibility issue that I described, there aren't multiple structures for membrane-bound enzymes. However, I don't think the answers will be much different for the membrane-bound ones. But we're actually, somebody else in the lab is looking at variants in membrane proteins. And that's interesting to see where the variants occur. There are extra constraints because of the lipid bilayer, etc. But fundamentally, the stereochemistry is the same, and the catalysis will be very similar, I think.
Okay, thank you very much. So the next question is from Johan Roer, and he said, what is your opinion on the feasibility of computationally predicting enzyme function data from sequence and structural information? The $64,000 question.
Challenge. And again, this is something that, in a way, one idea is that we could use this mechanism prediction to, with a new structure, then we could try and suggest reactions and see if we could find a mechanism that would work for it. So that's one of the sort of background ideas about predicting, or at least predicting possible mechanisms. I think of course, it depends. We all know that the binding is very sensitive to the energy involved, and often you have a very delicate balance between the binding of different molecules. That said, I think with the new techniques of taking a protein and doing crystal structures with many different small molecules will give us more insight into that. And I would hope that some of the machine learning methods might be applied to that, because I think for enzymology, the biggest question, we can often say this is a hydrolase or a serine proteinase or whatever, but we don't know exactly what the substrate is, and we don't know which biological role it plays in the organism. So there's a lot to be discovered there, and neither of those problems are easy.
No, I'm sure they're not. So thank you. So the next question is from Hector Rivaros Roses, and he said, do you have any idea why some protein superfamilies exhibit a great variety of functions with different substrates and mechanisms of reactions, like, for example, alcohol dehydrogenases and related enzymes, but other protein superfamilies possess very few reported functions, for example, pyruvate kinases?
So I don't know why. I would like to try and predict the evolutionary path, actually, but that's another problem again. What is clear is that it's partly evolution. So probably LDH, the tin barrel, all of these were present very early in evolution. And once nature has a stable protein structure with a binding site that is relatively flexible, then the potential to bind different substrates is there. The potential to perform catalysis is there. And nature will evolve things that work on the available substrates that are floating around in solution. And so it's partly a case of evolution. It's partly the structure of the bind. So, you know, things that evolve very early, I think, are often those that have many functions, etc. The structure of the binding site, if, as in a tin barrel, you have 8 loops that contribute to the binding site that are all relatively mobile, and then you can play, you know, you can play with them and develop new functions, whereas others just don't have that flexibility depending on the binding site. So that's a waffle. I mean, I honestly don't know the answer to that in detail, but it is a very, because, you know, about 10 or 11 different superfamilies constitute 50% of all proteins. I mean, it's radically non-normal distribution. It's an extreme value distribution.
Right, thank you. So we have some very interesting questions, I have to say. There are no easy ones, I think. So for next is from Natalie Bevin, and she says, given the overall structure of an enzyme can change so much, how challenging would it be to predict additional moonlighting functions of enzymes? And
well,
I think
that's an even more difficult one. And I honestly don't think, I can't see a way to do that, because that is purely evolutionary happenstance in my mind. And by moonlight, different people mean different things by moonlight. That's the first thing. So when I think of it as being one enzyme that does one thing in one place and a different thing in another place, or, you know, or at a different time or in a different environment. So it's the same enzyme. It's not like the evolution of the family where you have different members that do different things. It's much more that one individual molecule can do 2 different jobs. And sometimes, I mean, the glutathione reductases, is it, or transferases, that are in the crystallins, where, you know, normally they're an enzyme in the crystallin, in the eye lens, they're just stability proteins that create the right environment for the light. I'm sure that was just happenstance. This enzyme was around, it was produced in large quantities, and it could be used to, if you pack the lens, to create the dielectric constant that was needed.
Okay, thank you. And the next one is from Jim Warwicker. Are there catalytic templates that are not seen that you would expect to see, and are 20 amino acids enough?
Well, they aren't, because we've got all the cofactors. That's very clear. The cofactors provide that extra oomph, if you like, to perform catalysis that can't be done by the 20 amino acids. I always think of them a little bit, the cofactors, as nuclear warheads that can, you know, blow a molecule apart. So the 20 amino acids aren't enough, but there are only about 50 different cofactors, the organic cofactors anyway, and the metals. So even with those, the biological toolkit for catalysis is inordinately limited. I mean, this is what has fascinated me for years, the fact that with 20 amino acids and a few cofactors, you can do all of these different reactions. And so it's a tiny fraction, if you like, of organic chemistry space. And again, it reflects evolution. That's what we've ended up with. And it's like the fact that antibodies can recognize any molecule still amazes me. And it's the fact that these enzymes, proteins, can catalyze so many diverse reactions and do it so effectively.
Okay, thank you. So I think we'll probably finish up with the questions now with one more question, which is a more methodological question, I suppose, from Gangdeep Singh. And he said, can the mechanism prediction method be used in reverse? Oh, sorry, excuse me, I've hit the wrong one there. So can you please mention some tools for finding the rigidity or flexibility of enzyme cavities after running the molecular dynamics simulations? Are there such tools?
So the cavities, so Roman Laskovsky in my group, who was responsible for the project suite that you mentioned before, Colin, he has this tool called SurfNet, which will measure the volume of a cavity, and then you can compare that volume between different enzymes, especially within a family. There are other tools that, well, we've not used because we've used the SurfNet tool. The flexibility in doing the analysis, I didn't really show very much of Yanis's work on looking at these active sites and the flexibility. But that's very interesting because, of course, there's a payoff between being flexible and being efficient as an enzyme. And you really don't want your catalytic residues to be wobbling all over the place, because then the binding is pathetic and the catalysis is very, very slow. And so very often you find that these binding sites, the catalytic core is quite rigid. And you get the two-state enzymes, but it really often doesn't affect very much the catalytic residues. The binding sites change, but the actual catalysis part is quite relatively rigid. I'm sorry, I can't give you the tools, but there are lots of reviews on this, so I suggest you go and look at one of those.
Okay, thank you very much. With that, I think we'll probably finish up with your grilling now, Janet. Thank you very much for answering those questions. And again, thank you very much for your presentation. So I'm going to finish with some shameless plugs for the Biochemical Society. So all of you that are attending today clearly understand that the Biochemical Society puts on these webinars. I think we've put on over 40 in the past few years, and all of these are available through the Society's website and through the YouTube channel. Our next, it says getting a piece of paper to read it precisely, our next webinar is on drug repurposing, opportunities to create patient impact, on Thursday the 24th of February. So you can register through that, for that through the Biochemical Society website. And as I said at the outset, we welcome other topics that people might wish to suggest and other speakers that we could potentially invite to give these webinars outside of the awards system. And finally, I'd like to encourage you to become members of the Society. The Biochemical Society is the sort of premier society for molecular biosciences, based primarily within the UK, but certainly very international in its outlook and content. So I would suggest that if these topics interest you, then you can consider joining then becoming a member of the society in the future. So with that, I will let Janet go for her well-earned cup of tea, and I will thank everybody for attending, and thank the society and the behind-the-scenes people who've kept us running pretty well today, despite both Janet and my technical inabilities in this area. So thank you very much. Thank you. Bye, Janet. Bye.
Bye-bye. Bye.